A very good morning, all of you. It's my pleasure to welcome all the participants, speakers, organizers, and the management for today's International E-Conference on March towards Innovations in Drug Development and Discovery. I welcome you all, the eminent speakers and guests. So, Dr. Sundeep Chaitanya, Director of Research and Innovation, Department of Biochemistry, University of Cambridge, and Dr. Krishnamraju Ankireddy, Research Scientist at Bert Films, USA. So this is the first international conference organized by Mahati College of Pharmacy, completely conducted in a digital mode in line with social distancing norms due to COVID-19 pandemic. The theme of this conference is March towards Innovations in Drug Discovery and Development. So we are very proud to announce a very young pharmacy institution in the state of AP is organizing an international conference at the earliest of its inception, and we thank the speakers to be a part of it. And we also thank our management, without whose determination this will not be happen. And now I request the organizing secretary, Dr. Swarnalata Madam, to introduce Dr. Krishnamraju Ankireddy, research scientist at Bert Films, USA.
A very good morning to all of you, sir. I welcome Dr. Krishnamraju Ankireddy, sir, is today's speaker. He is a senior research scientist at Bert Films LLC, Kuteski, USA. His current research work involved in developing novel methods and materials for silicon solar cells. He's graduated with a master's degree in physics from Osmania University, Hyderabad. Then he moved to USA and pursued master's degree from Missouri State University, Missouri, USA. Later, Doctor of Philosophy degree from Material Science and Engineering from South Dakota School of Mines and Technology, USA. His 14 years of research career, he explored various types of nanoparticles and their properties for multiple fields such as printed and wearable electronic sensors and solar cells. Then he published the research work in many renowned international journals. He is also a trained and mentor for many graduates and undergraduate students throughout his career. Then he is always a passionate as a teamwork and collaborative with research efforts to give all the students, sir. Okay, sir, then you can continue.
Good morning, everyone. I guess for me, it's late night. In USA, it's 11:30 p.m. on Sunday night. Before I go to sleepy, I'm gonna get started now. Thanks for a nice introduction about me. My name is Krishnamraju, and I'm working as a senior research scientist at Bert Films LLC. It's a company that is mainly focused on developing materials for solar cell applications. So my responsibility in that company is mainly optimizing materials and manufacturing process to develop a particulate paste for solar cell applications. So I mainly lead R&D team to optimize and tailor the nanoparticles and micron size powders to make particulate paste that are helps to improve the efficiency of the solar cells so that we can reduce the cost of the solar. So that would be so that a lot of people can afford the clean energy. So with that background, prior to this job, I worked at several universities and worked with many collaborators on synthesizing nanoparticles. So I did worked on different types of nanoparticles, metal and semiconductor nanoparticles, and I use them in different applications such as printed and wearable electronics and some chemical sensors and solar cells. Today I am very passionate about sharing my knowledge about these nanoparticles. Of course, this is a vast subject. It's hard to cover within an hour of presentation. And I also chose like a broad title like synthesis and characterization. There are many ways to synthesize and characterize nanoparticles, but my focus today is to give some basic knowledge about the nanoparticles. But by the end of the presentation, I want you to take away some of the points about nanoparticles and how they are used in different applications, especially in a biomedical field. Without further ado, let's jump into the presentation. So this is the outline of my presentation. So I will start my presentation by discussing about what is nanoparticle and its properties, and then I will give a little bit background about nanotechnology, how it is utilized in different fields and even in um, biotechnology. And after that, I will discuss about synthesizing the nanoparticles. So at least I will discuss one of the routes, easy routes to create, fabricate or synthesize the nanoparticles. And once you synthesize, then you need to characterize the proper particles. So I will talk about how to characterize the nanoparticles with various techniques. At the end of the presentation, in the last section, I will talk about nanoparticle applications, how they are utilized in different fields. For example, printed and wearable electronics, and especially in biomedical field. So, with that, let's talk about nanoparticles. I know most of you know about nanoparticles. Some of you might have some research background in nanoparticles, but for many of the students, you may not be exposed to that much of the nanoparticle research background. So I want to give you some basic information about nanoparticles, and we will go from there. Um, so what's a nanoparticle? There are many definitions out there, but typically any particle or any structure that's one of its dimensions, if it falls between 1 to 100 nanometers, that is considered as a nanoparticle. So to give you a perspective of what is that scale, what is that nano means, so I took some one of the figure from the literature. From the top right corner, you can see um, so here, that's the scale. We are talking about nanometer. For example, if you take the water molecule, single water molecule, it's zero point 0.1 nanometers, less than 1 nanometer. And if you all know, since you have the biotechnology background, I try to tailor my presentation towards bio molecules, all that. So let's see. So here the glucose and that's in 1 nanometer size range, and proteins and antibodies are in a 10 nanometer range. If you consider the virus, that size is in that 100 nanometer range, and the bacteria is 1,000 nanometers, or in other words, you can say 1 micrometer range. If you are talking about cancer cells, they are 10 power 4 to 10 power 5 nanometers. That means it's about 10 to 100 micrometers. Think about your human hair. Human hair is about 18 micrometers to 180 micrometers. That is in that size range. So we are talking way much smaller than size than the human hair of this nanoparticle size. If you zoom, so our particular interest is in 1 to 100 nanometer range. Here is the zoom in picture. Let's see what nanostructures that comes under this category in 1 to 100 nanometers. For example, the gold nanoparticles, they are in the size in the range of 1 to 100 nanometers. There are quantum dots, meaning they are even tiny particles, 1 to 10 nanometers. Some are the metal oxide nanoparticles, and some of the liposomes and the polymer, and all these comes under 1 to 100 nanometers. So these are nanostructures or nanoparticles. So what is special about nanoparticles? Why we are so fascinated about nanoparticles? What's the difference from a small particle versus the bulk material? So there are some differences. There are some special properties for the nanoparticles. If you take the nanoparticle, for example, they are so tiny in that small size scale. So they have high surface area to volume ratio. Then what's the use of that means? For example, if you take silver block, right? So silver in bulk material, if you want to melt it completely, it needs about 900, 940 degrees centigrade. You have to heat it up to that level. But if you take the nanoparticle of the silver, if it is 1 to 10 nanometer range, the melting temperature of that particular nanoparticle goes down to 100 to 150 degrees centigrade. That means you are reducing the temperature for the melting temperature from way above 900 degrees to 150 degrees or 100 degrees, the workable range. So that's because of the special property of the nanoparticles. And there are many properties, but the other things are size dependent optical properties. Depending upon the size of the nanoparticles, the surface plasma and resonance, it's called, those are the responsible for the different colors they exhibit. So that can be utilized in bio labeling. So the nanoparticles have different colors, depends on the size. So these are all very useful properties that can be used towards developing biosensing sensors or drug delivery. But one of the challenges with these nanoparticles is they are so small that they have high surface area and surface energy. They try to combine each other. If you keep two particles close enough, they will try to combine each other and form bigger particles so that their surface energy will get reduced. So but when they combine each other, then they will be no longer nanoparticles. They will become micron-sized powders. So to prevent that agglomeration or the coalescence or the combining each other, there are two stabilization mechanisms are there for nanoparticles. So the two ways, if you see the right bottom figure, there are two stabilization mechanisms. One is called electrostatic stabilization. The other one is steric stabilization. So I will go through these two in detail. So electrostatic stabilization means you are using the electric charge to repel the nanoparticles each other so that they won't combine each other. For example, here the solvent has positive charge, and these uh, particles have the negative charge, and because of this positive charge cloud here, they will be separated in a solution. And the other mechanism that the researchers use is steric stabilization, meaning nanoparticle is capped with some organic molecules. So these organic molecules bind on the surface of the nanoparticles. They have some carbon chains, you see here, and when they come close to each other, these chains interact and they push away the nanoparticles. So that's another mechanism used to stabilize the nanoparticles, so they will be in the nano size range. Later, we will talk about how the antibodies will be connected or attached to these organic molecules for bio applications. But keep this in your mind that there are predominantly two stabilizing mechanisms for the nanoparticles. Why we need to keep them separate? As I told you that they will become bigger size particles, but there are other things we need to consider. I will talk in detail in the coming slides. So with that, so these are the nanoparticles. utilized in nanotechnology. Nanotechnology means the National Nanotechnology Initiative in USA defined as nanotechnology as if any research and development that involves structures or devices or systems in atomic or molecular level or macromolecules such as proteins. So that comes in the range of 1 to 100 nanometer scale. So if that technology deals with that, that is called nanotechnology. And this nanotechnology has numerous applications in many fields. For example, the left side chart, you can see I put at least some of the areas that nanotechnology is utilized. For example, coating and paints. In the paints, manufacturers, they combine these TiO2 particles and also the silicon dioxide particles to make these paints. And printed electronics, I'm going to talk more details later on how it is utilized in device fabrication. And then displays, for example, you all know that there are all the LCD TVs and the QLED TVs are coming, that is quantum dot LED TVs. Quantum dot is essentially a nanoparticle with 1 to 10 nanometer. So these nanoparticles are utilized already there. And then cosmetics, for example, the lotion that you use, some of the lotions, they have these TiO2 titanium particles to block the UV radiation from the sun. And of course, in catalysis also used these nanoparticles. In terms of biomedical applications, nanoparticles are used even in biomedical too. The detection and diagnosis of, for example, cancer cells, and the drug delivery and 3D printing. For example, the tissue cells are 3D printed to different shapes, and also imaging. For example, MRI. MRI works with a magnetic field. So magnetic nanoparticles like iron oxide particles are used researching to utilize in MRI. And then biolabeling is one more other application where the the emission or the light reflection from the nanoparticles are utilized to detect the targeted cells. So as you see, there are so many fields that are tied to nanotechnology, and also there is many applications even in biomedical field. Obviously, we are not going to cover all of them, but I just want to give you brief idea like what are the potential what is the potential for the nanotechnology and nanoparticles in various fields. So to give you a bit more motivation about nanoparticles, why those are important. So this is the slide. I did some literature review about how people are utilizing for making the biosensors. For example, every manufacturer, first their criteria is if they are making a device, is it cost effective? Is it low cost so that company can get profits, right? So ultimately, that's the hello. Oh, slides are not changing. I'm changing the slides here. Oh, I see. So is that is everybody is on a background page with the nanotechnology? Okay. 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 Sorry, I just got a message from the host that my slides were not changing. So I don't know what's the technical issue or the lag behind here and there. So as I mentioned earlier, there are many avenues for the nanotechnology. It can be utilized in many places, and I hope you are on same page now. With so people are utilizing in I was talking about the cost, right? The people or the company, any company, they are making products, they always look for the low cost, and so how they can manufacture the low cost devices. So one of the ways is to have large area printed electronics. For example, here in the figure A, you can see the nanoparticles ink is printed using the inkjet printer. So as the electrodes, every device need electrodes to monitor the signal, or so you need electrodes. So that electrodes are printed with uh, inkjet printer here, and later on they were sintered. I will talk about that in detail later. But here in the figure B, the nanoparticulate ink, these are nanoparticles utilized for printing on a flexible substrate. And then they further made devices for binding the antibodies and the proteins for biosensing applications. And then in the right side, figure C shows the glucose sensor, how they are making that. And the figure D shows the flexible glucose sensor, where they printed gold and silver nanoparticles for electrodes, and then they dried them up. I will talk about that, the sintering, all that process. But this slide, my intention was to show that there are several applications for these nanoparticles, as nanoparticles are printed in different forms. So with that, I want to talk about, I will come back to the applications later. So there are so many exciting opportunities for the nanoparticles. Now we need to know how these nanoparticles are synthesized. So with that in mind, in the next slide, I'm talking about how to synthesize the metal nanoparticles. So I did this research long back. So I synthesize the nanoparticles. Earlier I talked about the two stabilization mechanisms: static stabilization and electrostatic stabilization. So here we are utilizing static stabilization, meaning capping the nanoparticles with some organic molecules for the stabilization. For that purpose, we used carboxylic acids. Those are one type of organic molecules with head groups. These red ball and stick model, this one, and these red um, balls were the COOH. That's a head functional group, the oxygen. And then gray balls, and this is called carbon chain, and the white balls are the hydrogen. So it's C6 means it has 6 chains of carbon with a COOH head group, and the C8 means 8 chains, and C10 is a 10 chain. So those chains will have some impact on the stability of the nanoparticles if you are dispersing in any solvent or in any medium. So these are the ones that I used for synthesizing the nanoparticles. And if you take a closer look, what is happening in the atomic range here, for example, if you think about the like bulk material, if you divide into microscopic level and in nano level, there are atoms. Every material has atoms, right? So the silver 100 surface, they have them some crystal planes here. This is one plane and this is the other plane. And then when the carboxylic acid gets close to these atoms in the 2 nanometer range, and they will chemically interact with silver atoms, and they will form a covalent bond. That's where they are chemically bonded, so they won't come off easily. That's why particles are capped with these organic molecules. So with that background, I want to give you some details about how we did synthesis for the silver nanoparticles. So there are many different approaches, like top-down and bottom-up approaches. So many different methods, but one of the easiest method is chemical reduction method, meaning using all the chemicals and solution based method. For example, if you want to make nanoparticles, like synthesis of silver nanoparticles, here we use silver nitrate as a precursor and the N-butylamine as a ligand. You mix them and it will become one solution. And these are the carboxylic acid. Earlier I was talking about, these are the ones they will cap the nanoparticles. They are mixed with toluene, that's a solvent, that's a medium, and they are mixed as a solution too. When you combine all these solutions, um, and then you add the reducing agent to become a solution 4, and finally you get the nanoparticles. Let me go in detail what happens chemically inside when you add all these solutions. So this is silver nitrate, that is a metal precursor, where that's the source for the nanoparticles, you can say. This is Ag plus NO3 minus, and then when you add all these ligands, so this N-butylamine interact with Ag ions, and then they form silver alkyl ligand complex. Then adding the reducing agent means it reduces the ion. So the Ag is in the form of Ag plus, so it will give one electron to Ag, and the Ag become neutral, and these Ag atoms become a nucleation site. Once they become nucleation site, there are so many nucleation sites close by. As long as you have enough precursor material, those nucleation sites of 4 or 5 atoms together, they combine each other. They start growing a particle bigger and bigger until you run out of these silver ions in the solution. And once it forms to the nanoparticle, these capping agents will covalently bond to the particle. And finally, the nanoparticle synthesis will be done with this formation of nanoparticle with organic layer on top of it. And then once you get this particles in the solution, you will clean the solution. I mean, you will clean the nanoparticles, and finally you will end up with nanoparticles. So let's say you got the nanoparticles here, but you don't know exactly whether it's in pure form, there is oxidation, or are there any other materials along with it, and what are its properties, and what's the size of these nanoparticles. So you need to know all those. So to do that, you need to characterize the nanoparticles once you get these nanoparticles. So to do that, we did several characterization. We utilized some characterization techniques to find out the properties of these the synthesized nanoparticles. So I'm changing to the next slide. I hope you're all on this powder X-ray diffraction slide. So this is one technique to find out whether you got pure, since we are synthesizing silver nanoparticles. So we wanted to make sure we got the silver nanoparticles and not any oxidation or silver oxide or any other particles. So one of the cool technique is powder X-ray diffraction. The name itself says X-rays. So you will impart some X-rays onto the powder. These X-rays will interact with the atomic planes as I showed you in the earlier slides. And when they are diffracted from the crystal planes, and that will be collected, and you get the graph like this. So this particular 111, 200, 220, these 3 planes are representative of pure silver for the bulk material. From this experiment, it confirms that with that synthesis, the all pure silver nanoparticles were synthesized or created. But now, until now, we don't know what's the size of the nanoparticles, how big they are, because earlier we defined the nanoparticles in 1 to 100 nanometer range. So we need to check whether they are in that range. So to do that, there's one more technique called transmission electron microscopy. The name itself suggests that the electrons are transmitted through the sample. So you have a carbon grid and you put sample on nanoparticles in a solution on top of that. The electron beam pass through that, and that will create an image. Here you can see these are the tiny dots are the nanoparticles. So this is the 50 nanometer scale, and some of them are 3 nanometer, some of them are 5 nanometer. The zoom in picture here shows that they are spherical in shape. Excuse me. So they are spherical in shape. So we did the size distribution diagram. So most of them are in that around 4 nanometer range. So this experiment, this characterization technique confirms that the nanoparticles are spherical in shape. They are in about 4 to 5 nanometer in size. That means we are successfully synthesized nanoparticles that are below 100 nanometers. There is one more technique that is used to see what are the optical properties of the nanoparticles. This is called UV visible spectroscopy. It is meaning that you are sending the UV visible light to the solution of the nanoparticles. So because depending upon the size of the nanoparticles and the material, it absorbs some of the light. Here you can see, the visible light is from 300 to 750, 800 range, right? So these particles are absorbing. This is the y-axis is absorbance and x-axis is wavelength. You can see most of the absorbance is high, around 420 nanometers. That means around 420, 400 nanometers, it's a blue region, right? If you go to 700 nanometers, that's a red region of the whip jar like color. So these particles are appear as a blue in color, and they are absorbing the light around 420 nanometers. So that tells that these are having the surface plasma and resonance that's at 420 nanometers, and they are spherical in size. So from all these techniques, what we learned is so we know these are the pure silver nanoparticles from the XRD analysis. And from transmission electron microscopy, we were able to see the size. They are about 5 nanometers. They are from 3 to 5 nanometer, very narrow size distribution. And to know the optical properties, the UV visible spectrum revealed that these are blue color particles, and they are absorbing the wavelength of around 420 nanometers. So this tells that we successfully prepared the nanoparticles, and then after you make the nanoparticles, then how you want to use it, what's the use for those nanoparticles. In the earlier slides, I showed you some flexible device glucose sensor that was some of the electrodes were printed. So our research was focused on using those nanoparticles and make the nanoparticulate inks, means mixed in a solution, then use the printing methods, different printing methods to print them for making the micro electrodes for the device. For printing, there are several printing techniques available out there, but we used a couple of them. Here I show you one of them is the Optomic aerosol jet system, and then Sonotech spray coater, and the inkjet printers. So let me give you in detail why we need nanoparticles. Why can't we use a bigger size particles for printing these patterns? To answer that question, let's talk about this aerosol jet printer. So here in that image, so that's the top, and if you zoom in, so here there's a nozzle where the ink or the nanoparticles come out and print on the substrate or any plastic material that you want to print on. So the mechanism that involves in this printing is, in the print head, what happens here is when the nanoparticulate ink enters into the print head, and this sheath gas, the gas will be introduced here, and they will form droplets of like nanoparticle droplets, and they have to pass through this nozzle. These nozzles are about 100 micrometer in that range. For example, if you imagine if there's a bigger size particles here, and those bigger size particles can be traveling through this nozzle, they will clog the nozzle and you won't get any printing. So that's the one reason for having the small size nanoparticles here. The other reason is, as I mentioned earlier, comparing to bulk material, nanoparticles have low melting temperature. For example, if you want to print on a flexible substrate like a plastic substrate, you can't heat the plastic substrate to 900 degrees. It will melt. Some of the samples that we use is a PET, polyethylene terephthalate. That's a one polymer film, so it cannot be heated more than 150 degrees. So you have to have the particles that should be very small in nanometer range, so that you print and heat them. Why we need to heat them? Because when you cap these nanoparticles with organic molecules, they won't be conductive anymore, because those organic molecules are not electrically conductive. You need to remove those organic molecules once you print on the substrate, so the nanoparticles combine each other and form a solid electrode for conducting the electricity. So we use the steric stabilization to suspend the particles in the ink that we needed. So that's the primary purpose. Once they are printed, we need to remove the organic molecules so that the particles combine each other. It's called the sintering process. Sintering means combining two or three more nanoparticles through the heat or any other means of different methods. And so the other mechanism is, that's for like a precise micrometer range electrodes. If you want a large scale area printing, for example, thin films, in that case you need spray coating. So we utilize the spray coating technique to spray the nanoparticles to form films on different substrates. I will show you some of the pictures later on how we printed those. So these are the two techniques in the early stages of our research. We use those. As I talked about the nanoparticulate ink, like I said, those are the nanoparticles capped with the organic layer. They need to be dispersed in some of the solvents, that's mediums. Here we mix these nanoparticles in a solvent called toluene, and when we mix that one, it has a uniform dispersion of the nanoparticles in the solution. So this can be used for printing, even in the inkjet printing. Instead of toner or ink cartridge, you replace that cartridge with this silver ink. So instead of black ink or other color ink, you are printing the silver nanoparticulate ink. So for inkjet printing, people use this kind of ink for device fabrication for the electrode printing. The other application, we also synthesize zinc oxide nanoparticles. Those zinc oxide nanoparticles have many applications. The one example is they are used as a transparent conducting electrodes, and also in terms of biotechnology, their antimicrobial properties are there for them. And also they are excellent UV blocking properties they exhibit. So there are a couple of advantages for even bio industry too. And so our goal was to print a transparent conducting electrode. So the ink should be transparent. If you see that 5 mole % of zinc aluminum is doped to the zinc oxide, those were cloudy in color, but pure zinc oxide were almost clear in color. And that graph, it shows that the absorbance, that blue curve here is 5 mole %, the cloudy solution where the absorption is more. But if you see other ones like a black line here, that's a pure zinc oxide, the absorbance is almost negligible throughout the visible region. So we were successfully able to create these inks, nano inks for device fabrication. For example, so after that, we use those printing systems for printing micrometer scale um, lines. Here, these are the electrodes with a different gap between them, and this is the other electrode with a 10 width, and the particles were printed close to each other. And so these are, like I mentioned, these nanoparticles are blue in color. That's what you see here. So these were not sintered, means they were not heated. So there's still these nanoparticles are having some organic molecules on it. So to make it conductive, you bake them or at 100 degrees or 150 degrees. In our case, we did it 150 degrees. And what happens is that in the next image you see here, excuse me, this is scanning electron microscope. You see these particles combined each other and it became more like a bulk structure. So that way the electrons can easily pass through that. It more like behave like a bulk silver metal. Some of them, this was printed on a Kapton substrate. It's a polyimide substrate. That's a polymer substrate. It's a flexible substrate that can withstand to up to 300 degrees. So these are the microelectrodes we printed on that one. And if you zoom in, what is happening in this printed region or this sinter region. So here, when we used a small, low weight percent of the nanoparticles in the ink, the printed pattern was not smooth. Our goal was to create these microelectrodes, but there's a lot of white space here. They sintered the particles. They are not in nanometer they are not in nanometer range because they are sintered. Um, and here, with higher nanoparticles in the ink, you see that this much more uniform or in less white space. It is much more conductive. So that nanoparticles concentration in the ink also plays a crucial role. And this is one more image. It's a cross-sectional image. After sintering, you see how thick they are, around 4 to 5 micrometer range. And they are very dense structures. The other one I talked about, zinc oxide nanoparticles. Our goal was to make a transparent conducting electrodes. So we played with the different solvents, and you see there were cracks, and some of them are whitish in color. But using a right solvent is necessary to get a uniform coverage. These are all optimizing the inks and nanoparticles and also solvents for the printer requirements. And even after getting, we were able to get a transparent film on the glass substrate, but still there were cracks after sintering. So we had to use some other additives called aminosilants to not to get the cracks in the films, and at the same time we get the transparent conducting electrodes. So the point of this slide is the micro electrodes that you see on for any devices. There's a lot of things needs to be optimized, or in the printing as well as the materials, nanomaterials, and also the process parameters to get your right structure and right conductivity for utilizing in any device applications. These are some of the scanning electron microscope. You see, without adding the silane, it was more like cracks, but adding some silane, it was uniform film was formed. And even the printing techniques also have some role here. What kind of printer you are using, what are the print parameters. Using one print technique, it was much smoother, but using another print technique, it was cracked film. So these are all the important optimization process that one has to undergo to get a uniform electrodes for the device fabrication. And finally, we were able to get uniform films. This is the glass substrate here. This particulate film is the one with these are the zinc oxide nanoparticles. I hope you guys are able to see my slides. Hello, Syed. Can you see my slides? So I hope you are able to see that. From my side, it looks okay. I don't know why that lag is, but from my side, it looks okay. It looks like you guys are now able to see the slides. I was talking about the different thickness films we were able to make for the electrode fabrication. So the ultimate goal of this research and using the nanoparticles is how we can make different electrodes and with a low cost, meaning that printing on flexible substrates and without much material wastage. So with all this, we were able to make electrodes, and we were also able to demonstrate some devices. Some of them, I know this is different for the printed electronics, but for example, we were able to print flexible antennas. If you see the top right corner, the B is the antenna that was made with a milling technology, traditional milling and etching process. So that takes a lot of material wastage. But if you take the left one, that figure A, it was printed with the nanoparticles that we synthesized and sintered them, and this is flexible. And if you see the graphs, these two graphs, the black one curve is the printed antenna and the red curve is the copper antenna. So the performance is almost close to each other. With less cost, we were able to produce this one. In addition to this, I want to talk about a little bit about how they are utilized in biomedical applications. Obviously, I'm not from biomedical background, but I try to show some of the applications that is used in biomedical application. As all you know, that ELISA technology, it's called enzyme-linked immunosorbent assays. Those are the test strips you usually get anywhere that are used for testing the proteins and antigens. So this particular test strip is used. These are called lateral flow immunoassays. So the point here is in the clinical sample with the analyte is placed on the sample pad on the top figure. And on the next strip here, the antibodies are attached to the gold nanoparticles. The important point is the nanoparticles, gold nanoparticles are utilized here. These are loosely bonded to this material, not covalently bonded. When this analyte interact with this one, with the capillary help of capillary flow, they will travel to the test line where these antibodies are attached to the test strip. It's a nitrocellulose here. And it will so in the second image you can see that these are traveling to there after attaching that analyte to the antibodies. And then when they travel close to this or travel through this test strip here, test line, these antibodies will be attached to the antibodies that is already on the sample in a sandwich structure. So this is antibody, analyte, and then antibody on top of that nanoparticles, gold nanoparticles. When when these reach and form this sandwich structure, on the test line you will see visible red line. that means the test is positive, and there's a control line here. The point here I'm talking about is the not only in the printed electronics, they are used in many fields. In biofield, these nanoparticles, gold nanoparticles, are used. Gold is chemically inert, and is much safer to utilize them in this immunoassay devices. So that is one application, and I want to talk about other one. So this is from another literature study, Romeo et al. They printed a glucose sensor that is especially utilized for sensing the glucose from tear drop fluid. Basically, it's a non-invasive technique. You are using the tear drop and utilizing that glucose if there are any glucose levels in that. So to do that, they use the inkjet printer here. As I talked about the inkjet printer, so they put the ink in the nozzles. For example, in this case, they used Fujifilm Dymatics printer, and that has a 16 nozzles, and then they place so they use these flexible substrates. This is called PET, polyethylene terephthalate. So that flexible substrate is used for printing, and then they use gold nanoparticles to print the working electrode and counter electrodes on top of this film. And also they use silver nanoparticles. They put it in the cartridge of that inkjet printer, and that will print on the substrate. And they used silver as a reference electrode. So the problem is, now once you print those, for printing you need to have the particles suspended in the solution. That's why you use the capping agent. But once you print those, since they are not conductive because of the organic layer, you have to sinter them or heat them to remove the organic layer. So what they did is they heated the structures at 130 degrees centigrade for 20 minutes. So that helped them to sinter the particles, or the particles come close together and they became conductive. So once they've printed the electrodes, they also printed some other layers, and especially they functionalize these electrodes with the copper oxide particles. The copper oxide is more sensitive to the glucose. If you see the working mechanism of this glucose sensor, copper oxides will interact with hydroxides to form copper hydroxides, and with one electron. And this copper hydroxide with electron interacts with the glucose, and it will change back to copper oxide, and it will create the gluconic acid. Depending upon the level of the glucose inside the teardrop, then you will get the signal. They use the cyclic voltammetry. That's one type of technique to detect the signal. So they did, for example, this graph here, current versus glucose concentration. So here, without copper oxide nanoparticles, the signal was low, but with copper oxide on the electrodes, it's more sensitive, and they were able to obtain the signal. So there are so this is one type of method people are using, or innovative techniques to do the large scale printing, and so the material cost and the production cost will be low, so they can do the mass production for the biosensing applications. And the other application, it's about cancer cells detection. This is from Georgia Tech University, where the antibodies are attached to the gold nanoparticles, and they were those gold nanoparticles with antibodies are sent to that targeted cancer cells, and the receptors will um, help to attach these antibody binded gold nanoparticles there. with the light that is reflected from these nanoparticles by shining the light on those, probably the IR light, you will get the imaging of those cancer cells. The point here is to show that these nanoparticles have the applications in even detection of these cancer cells, for the diagnostics purpose. So like I said, even though I don't have the biomedical background, but these three from the literature, you can see there are ample opportunities for the nanoparticles because of their size. They can be utilized in many fields. With that, I would like to conclude my presentation. As you see that these nanoparticles are, even though they are tiny, but they are finding applications in numerous fields because they have special properties comparing to bulk materials. And also we discussed about nanoparticle synthesis. Especially I explained about one of the chemical routes to make the nanoparticles and how to stabilize the nanoparticles. And then also I gave you a brief idea about how to characterize the nanoparticles, what type of characterization methods are available. Then once you create the nanoparticles or synthesize the nanoparticles, how you want to utilize for the device fabrication, even bio applications. So for that, we discussed about flexible printed antenna for printed antenna applications, printed electronics applications, and then glucose sensors and cancer cell detection for bio applications. I hope I gave you some basic idea about the nanoparticles, and I hope you got some idea about some of the cool applications of the nanoparticles and how they can be utilized in bio industry. With that, I would like to conclude my presentation, and thank you all for giving me this opportunity. I think I'm done, and if you have any questions, feel free to ask me. Thank you.
Sir, we had got a question from some of the audience.
Sure.
Sangar asked, yeah. Sir, what happens if we inhale these titanium particles? They have asked the question, titanium nanoparticles, regarding to the titanium nanoparticles. What happens if we inhale?
Good question. The titanium nanoparticles are mostly benign, and as you mentioned, the nanoparticles always they have some toxicity always combined with them. But the especially these titanium nanoparticles, they are TiO2 nanoparticles are utilized mostly. In the lotions, body lotions, they are in the range of 10 nanometers to even some of them are 100 nanometers. So they don't pose much threat to the health, but I'm not sure how the nanoparticles will interact with the lungs. There might be some effect there. There will be some effects with all the nanoparticles, but I'm not particularly sure how that interact with the lungs. But all the nanoparticles, you should be very careful when you are dealing with the nanoparticles. For example, when we made these silver nanoparticles, we always work in the hood, fume hood, and so that the aerosols or whatever nanoparticles come, you won't inhale those because they easily can penetrate through the skin membrane. That's the difficult part to take those out.
It's a great presentation by Dr. Krishnamraju Ankireddy. We are very happy for sharing your knowledge and research experience with us related to nanoparticles. And thank you so much for giving us your valuable time regarding to educating regarding to the nanoparticles. So now I request Dr. G. Swarnalata Madam to introduce Dr. Sundeep Chaitanya sir.
A very good morning, sir. Dr. Sundeep Chaitanya is working as the Director of Research and Innovation at the American Leprosy and Research Associated at the Department of Biochemistry, the University of Cambridge in the UK. He graduated and postgraduate degree from Andhra University with distinction. Now, he did PhD in molecular biology and immunology of leprosy from India. In 12 years of laboratory-based research experience in genomics, transcriptomics, immune biomarkers, and computational biology, Sandeep sir leads multi-centric scientific research projects in during drug discovery and development of diagnostics for leprosy, with a strong passion to translate laboratory research to reduce the global burden of leprosy. Now, Sandeep sir is serving as a principal investigator on 3 projects. The projects are funded by American Leprosy Mission at the University of Cambridge and Leprosy Research. Now, the Sandeep sir is the member of Technical Advisory Group in Diagnostics in Neglected Tropical Diseases at WHO. Sir, now I request to give a topic on structure-guided fragment-based drug discovery for leprosy.
Thank you very much, Dr. Swarnalatha, for the introduction. Yeah, am I audible? I think I would request the host to speak and let me know if I'm audible and if my slides can be seen. I'll share my slides and yeah. Can all of you see my slides now? Maybe if host can respond, that would be good.
Yes, sir, slides are been visible.
Yeah, okay. Thank you. Sorry for the background noises. I'm currently in India for the next 15 days. I'll be in southern part of Andhra Pradesh, and then I'll be heading to Vellore to do a DNA sequencing project on drug-resistant strains of leprosy. Let me introduce myself. I'm born in Narsapur, which is in the West Godavari district. I speak Telugu with many of you who have joined this session today. I've done my post-graduation at Andhra University. Then I did my PhD in molecular biology and immunology with a research center that's connected with the Leprosy Mission Trust India and a university in Northeast, which is called as Martin Luther Christian University. So it's an industry-sponsored PhD program where I did my PhD, and then moved to Cambridge to do my postdoctoral studies in basic bioinformatics and computational drug discovery, which is for 1 year. After that, I have moved to be the research associate at the University of Cambridge, leading the structure-guided fragment-based drug discovery program to identify novel therapies for leprosy. So that's the background. Just to introduce leprosy as a public health problem. Leprosy continues to be a significant public health problem in endemic tropical countries. Many of you might have heard about leprosy, which is a debilitating disease similar to that of tuberculosis, caused by a bacteria called Mycobacterium leprae. This disease disfigures the humans with the clawing of the hands, the loss of sensation, patches all over the body, and it's a very complex disease which stigmatizes a person's life from the time of infection to almost death if the deformity occurs. Many people have an understanding that leprosy has almost gone from the world, so it's no longer a problem. But there are almost 200,000 new cases detected each year around the world, especially from the 159 WHO member states and territories. As you can see the map here, Brazil, India, and Indonesia seems to have the highest number of cases, of which India reports the highest number every year, approximately 120,000 new cases. There are 23 countries reporting around 9,795 cases with visible deformities. So these are the people with real clawing of the hands, loss of vision, and there are also situations where there are the loss of limbs, especially the toes and the legs. one of the biggest challenges with leprosy now is the evolving antimicrobial resistance. There are only 3 drugs known to act very well on Mycobacterium leprae. That is dapsone, rifampin, and clofazimine. These are the 3 drugs that kill the bacillus, and they're the part of the multi-drug therapy that is used to treat Mycobacterium leprae. Resistance started to evolve for dapsone a long time ago, then for rifampin, and ofloxacin, which is actually a second-line drug, which is also showing resistance recently. So there is no serological test or a good genetic diagnostic test available so far. The diagnosis is primarily clinical. There is no vaccine to prevent leprosy. BCG has been used in few countries, but the protection is just 30%. Duration of the therapy, the current multi-drug therapy is for 1 year to almost sometimes 5 years in few countries, which has caused quite a lot of issues with the compliance and the increase in defaulting rates. Drug resistance has been evolving. There are a lot of reports in the last 10 years from different countries, and the transmission is ongoing, and there's quite a lot of susceptibility among the contacts of leprosy cases that are noted across the endemic tropical countries of the world. I would just introduce leprosy as a disease initially. As you can see, Mycobacterium leprae, the bacilli that causes leprosy, is a acid-fast bacteria that can be stained by only counterstains. So it has got a thick waxy coat that contains very complex mycolic acids that do not let any of the gram stains penetrate. So it needs to be stained with a counterstain, which is a Ziehl-Neelsen stain, we call it. So it's a acid-fast bacilli. The mode of entry of Mycobacterium leprae is largely through the nose. We don't know if there is a direct contact still, but it's known to be largely through the nose. Mycobacteria is exhaled by an infected person. It goes into the air, it forms droplet nuclei in the air, and then when another person inhales, and if their immune system compromises, then they can actually develop the disease. Disease is largely classified into paucibacillary and multibacillary disease. The paucibacillary disease is for 6 months, which is noted by very few number of skin patches, as you can see here, towards the TT side. Whereas the multibacillary disease is a diffuse form which occurs throughout the body and needs a more rigorous, lengthy treatment. This is the field-based classification of WHO. But later, then the immune spectrum around which the disease is distributed has got 2 extreme poles called tuberculoid and lepromatous. The TT represents tuberculoid leprosy, and then the LL represents the lepromatous leprosy. And these 2 forms are actually separated by borderline groups called borderline tuberculoid, borderline or mid-borderline, and borderline lepromatous. So the disease usually starts from a TT phase and slowly progresses towards the LL phase. And the immune system also varies quite a lot, as you can see in this slide. One of the other challenges with leprosy is the sudden reactions that occur during the course of the disease. Suddenly, an immune reaction can occur, which directs the individuals into a state which is highly responsive to any external agent, and then it sets in an aggressive delayed type hypersensitivity reaction, which can sometimes lead to death. So these are the major complications of leprosy, and it has continued to be a significant health problem to the tropical world. Looking at the genomic features, before even getting to the genomic features, there are, as I have shown to you in the earlier slides, there's quite a lot of research needs that exist for leprosy, mainly due to the lack of a good therapeutic which can actually treat the disease in a short duration of time, say like in 1 to 2 months, and kill and clear the bacilli from the host. Secondly, there is no good vaccine. We don't have a specific vaccine that can act on leprae as that of tuberculosis. So for tuberculosis, BCG has been used and is being used for quite some time, but for leprosy, we don't have a vaccine like that. There are few vaccines in trials. I'll come to that later, but as of now, there is no approved vaccine for leprosy. So all these gaps or lacunae in the understanding of the disease and how mycobacteria infects the host, Mycobacterium leprae infects the host, has been a challenge for quite some time. The added challenge with Mycobacterium leprae is that it cannot be grown in an axenic culture media like that of any other bacteria. So it doesn't grow on any media. It just dies if we put a culture or if we put an extracted bacteria onto the culture plates. So the only mode to propagate this bacteria is to put them in the live animals. So mouse foot pads, the hind foot of mice, are injected with this bacteria, and it takes 6 to 9 months for the bacteria to grow and cause infection in the mice. So that's the only laboratory-based infection model that is available for the scientists to study either the impacts of drugs or the biochemical and metabolic characteristics of the pathogen. So the genome of this bacteria, Mycobacterium leprae, has been deciphered in the year 2001, where they recognized 1605 genes. It has recently extended to 1615, but this is the original reference genome, which is called as Tamil Nadu strain. This strain has been first identified in Tamil Nadu, which is from the footpads of crossbred albino mice from a center called Karigiri in Vellore, where I'll be visiting in the next week. So when they identified this strain, they recognized that these are the characteristics of the genome. There are 1605 genes, 45 tRNA, and there are a significant amount of pseudogenes. These pseudogenes are actually functional homologs in tuberculosis. So that's a confusing part with this bacteria, is that it has slowly and reductively evolved. In the sense, it has shut down the activities of many of its genes and still managed to survive in the human body and cause disease for many centuries now. So the other genomic features you can see here, of all these, the interesting piece is the number of structures within the protein data bank to understand the bug's proteome. It's such a strange situation that for tuberculosis, if there are around 2,000 gene protein structures, like gene product structures solved, there are only 12 structures available in PDB for M. leprae. So this warrants or raises a question that not much of structural biology has been done for leprosy, and not many structural studies have been done. So drug discovery has been limited in the past to only animal model experiments. So there are new drugs given to mice, or maybe repurposed drugs that are administered to mice, and the impact of the drug on the bacilli is studied through in vivo responses. Because there is no in vitro model to study, or there is no basic structural studies done so far. Like, very limited number of studies were done so far. You can just see 13 to 15 publications in PubMed related to structural aspects of leprosy genome. So that's a challenging thing to me, because we need to really look for novel therapies. Otherwise, we are going to just end up with a long duration therapy, wherein secondary complications can set in the patients that lead to deformity, and also it contributes to the emergence of drug resistance. And if drug resistance occurs to these primary drugs, then there is no other alternative available so far to really aggressively treat leprosy and cure patients. So with this idea, I have been to met this person, which some of you may have known, because he's been a pioneer in drug discovery, cancer drug discovery, and mycobacterial drug discovery. I met him at a conference in 2016, and we were talking about Mycobacterium leprae. And then he called me to visit his lab and stay and work for a short project. But the short project ended up as a much longer project, and almost now a permanent position for me to settle in this department. The building that you see behind is the Biochemistry at the Central University of Cambridge, under the central office of the University of Cambridge. So at Biochemistry, Tom has started his work with Professor Sir Tom Blundell. As you can see, there is a Wikipedia page for him. And I work now with him directly in his lab, leading this leprosy drug discovery group. Tom has been on the forefront of drug discovery from the time where he worked with Dorothy Hodgkin and Rosalind Franklin on the earlier structures of insulin. He was in the group that solved the structure of insulin, and I think that's the time he started to think about writing programs to model the structure of the proteins, because proteins have similar structures, although they are distant in their homology. Although their functions are different, although they have a long relationships, they still have similar structures because the folding has got a pattern, because we are just limited with 20 amino acids and few modified amino acids in nature that we know of. So with these very initial thoughts and ideas, his first program, which is Comparer, then developed as Modeler in 1993, has been on the forefront of bioinformatics groups around the world, with around 15,000 citations, is what he says last week. It's his amazing work, and his group, Professor Andres Shali and other colleagues who now set it up at the University of California in San Francisco, where they were continuing this project on Modeler. I think that's the initial days in which he really looked at the structure of the proteins and their relevance to function and understanding of small molecule binding that can lead to eventual and potential drug discovery. So he has this original ideas on developing programs, computational programs that aid in developing modeling proteins if there is a challenge in actually solving the structures of proteins in an experimental design or an experimental fashion. So there are a lot of programs that were developed in his lab. I can share these slides, and if somebody wants to really go and have a look at these programs, they can. But the interest of showing these slides is that originally the programs were used to model the structures of the proteins, understand how they align with the homologous proteins in the space that are known, and then there's quite a lot of work done around understanding the impacts of mutations on the structures of the proteins, or maybe in small molecule binding. So that has led to an excellent work done with about 50 papers in Nature for him, related to cancer biology and understanding the impacts of mutations on cancer. In the year 2000, Bill Gates has stepped into his lab. I don't have the pictures of all of them, but Gates has stepped into his lab and asked him to think about Mycobacterium and the problem that it causes to the world. So 2001, with the first Gates-funded grant, he started to understand the genome of Mycobacterium tuberculosis. Quite a lot of drug discovery has been done. I would be proud to say that there are at least 2 drugs of his research have been on the market in the forefront. And most of you know that the pathway of the pipeline to drug discovery is quite challenging. It's not really quick as we think of, because there are trials, there are validations that will take more than 20 years for each drug, sometimes to get into the market, which is now being spearheaded with advances in technologies, but that has been the timeline since then. So he has put at least 2 drugs in the market, and there are quite a lot of drugs on trials and basic research in single cell experiments on animal models and other things. So from then onwards, Mycobacterium tuberculosis research started in his lab. In 2012, he started working on Mycobacterium abscessus, which causes cystic fibrosis and other pneumococcal infection, or other pneumonia-related infections in lungs in the patients in the UK. So he started to work on Mycobacterium abscessus as well. In 2016, I met him and I interested him of Mycobacterium leprae, and then we started to work on Mycobacterium leprae drug discovery, trying to understand the proteome of leprae and identify small molecules that can actually bind with some of the proteins of leprae and inhibit their function and kill the bug ultimately. The objectives of my project are to really model the proteome of Mycobacterium leprae, model the entire proteins in the proteome of M. leprae. The modeling that was developed in the lab is quite a complex process. The reason is that we don't really depend on single template modeling. We really love to do multi-template modeling covering the entire query sequence that we model, and also identify right folds for the regions which otherwise do not have in other templates, and which depends on single template modeling. And also, the systems that were developed in the lab can model not only just the proteins, but also small molecules within the proteins, so that it gives a more comprehensive understanding of how the protein can look like even before it is solved experimentally. We would like to put it as a database, which is under construction. I will talk into details of it shortly. Then it also interested me to develop something called as HARP, which is a database of impacts of systematic mutations on different drug targets in leprae. So if we have dapsone, rifampin, and ofloxacin. We have 3 known drug targets for that, whose structures have been solved in TB. They are highly homologous in the sense they are highly identical to the proteins in leprae. So we built the models and we introduced impacts, and we introduced mutations and measured impacts of all these 3 drug targets in leprae. We developed a database named HARP, which otherwise means Hansen's disease antimicrobial resistance profiles. Hansen is a scientist who identified Mycobacterium leprae bacillus in the year 1973. He's a Norwegian physician who identified this bacilli. So Hansen, the database is named after him, and HARP, which also means Hansen's disease antimicrobial resistance profiles, is named after him. And you can also see that some of these carry the names of the musical instruments, as you would have seen in the slide on which I'm introducing Sir Tom. Sir Tom has been a musician. He's been a jazz player for many years, and from there, Dorothy Hodgkin has actually interested him into science, into structural biology work. So he moved into science, but music hasn't left him. So most of the databases we build in the lab have some type of musical name with relevance to the work that we're doing. Then the third objective is to systematically prioritize drug targets from Mycobacterium leprae and conduct structure-guided fragment-based drug discovery, which I'll be talking in detail in the next slides. So for Hansen, we started to characterize the genome using all the computational tools that we have in hand, looking at the gene ontology annotations, having to have any structural features that are documented either in UniProt or any other databases. We also used a multitask protein modeling pipeline, which is in-house, which hasn't been published yet. It's still used in-house. That depends on CAP and SCOP classification of proteins. I won't really go into details of these, but these are all available online in published literature. You can browse through and learn from them. So this pipeline actually helps to model the protein from its sequence to the structure. So it's completely automated multitask pipeline that's run on the supercomputers here at the University of Cambridge, where we can submit a proteome instead of even a single sequence, and it can model this proteome in few hours of time using multiple templates and generating various small molecule binding states to each of the proteins. So for say 1615 proteins, I have generated around 128,000 models, which are multiple for each gene, as you can make out. We are in the process of now prioritizing the models using different metrics, parameters, and then putting them into a database called Hansen. So apart from just understanding the structures, on the other side, you can see in the green boxes there, we are trying to look at the essential genes, because ultimately the idea is to understand the structures of these proteins, solve structures of some of the proteins, which are highly essential for the bacteria to survive, and block their function with small molecules, the potential small molecules. So we use different approaches to determine the essentialome, we call it, which is the essential genome of the bug. There was transposon saturation mutagenesis experiments done in tuberculosis. We took those transposon sites, extrapolated them to leprase genome, and then we are trying to map the essential genes. We are also doing the expression profiling. We did transcriptomic studies at Vellore from the M. leprae that's extracted from the foot pads of mice. We did computational choke point and metabolomic analysis, and close to determining the druggable proteome, which we are naming it as drugulome at this stage. These are some of the terms that we are coining as we go by. And then we also look at some of these epistatic interactions, the impacts of mutations. What are the potential small molecule binding sites on these proteins, like the fragment hotspot maps? And then if there are any epitopic features, because that can provide some level of insights to the diagnostic development for leprosy, which is also a huge need, as Dr. Krishnamraju was speaking. ELISA could be an excellent format for detecting antigenic responses to M. leprae, Mycobacterium leprae, but we need to have highly specific epitopic regions that are structurally characterized to have this designed for leprae. So to develop diagnostics, we are actually putting in this information as well into Hansen database. So far, we managed to model 1,428 proteins out of the 1,615 genes. There are quite a lot of small molecule bound, unique small molecule bound models. There are different models developed in apomeric states and states bound with small molecules. But of course, these are the refined set of models, I would say, from the large chunk of data that we have collected with the modeling pipelines in-house. At least we managed to do around 80% of the proteome with best quality. There are a few models that are of poor quality which might be removed from the process, but that's the way forward to have a quick understanding of the structure of the proteome of M. leprae without having to do exhaustive amount of exercise, structure solving exercises to solve all the proteins of the proteome. So yeah, as I was talking, Hansen is under development. We are in the process of developing it, and this will encompass all the 3D models. It will also provide information on the protein nucleic acid, and protein ligand interactions. It enables researchers to identify the druggable proteome very quickly. So that's the idea of developing Hansen. The two large objectives of these studies are to not only just do structure-guided drug discovery ourselves, but also provide these tools for the research community around the world to use the knowledge and generate their own studies, or having to even analyze the data that's put into some of these databases and use it to make meaningful publications for the future. So this is in development, and this will be shared shortly, but HARP has been published and it's available online. These are some of the steps that we use to do the drug target identification. It starts with having to have an understanding of the gene ontology and do some choke point analysis to determine choke point reactions, which are essential for the bug to survive. Then we go to the gene level, understand the homology, understand the essentiality by homology, and also, if possible, do some level of transposon saturation at the in vivo level, not at the in vitro. For this, we actually started a small project recently with our collaborators in Vellore to introduce CRISPR-Cas9 into the foot pads through in vivo transfection and edit the genes so that we know the essential component of the genome of Mycobacterium leprae. So this project is undergoing, like it's in process now. We'll have some findings maybe over the next year or the following year. We also looked at some basic mathematical models, mainly the flux balance analysis. We took the gene expression data that we have generated if some of you are interested in this, you can go to the Gene Expression Omnibus repository of NCBI, and you can browse through with my last name, and you should be able to get these experiments. All of these is downloadable. One can have a look at the experiments and how we are analyzing it. The other important challenge that we normally face during target identification is the homology to human proteins, and also homology to human gut microbiota proteins. So we tend to avoid that using different tools that were developed online. We write our own programs, and since this is a programming lab, and we run them to check non-homology with human proteins and also non-homology with human gut microbiota. Then we go to the structure analysis. If it is possible to really solve the structure of the protein experimentally, we do that using either X-ray crystallography on the diamond light source, which I'll be showing you shortly, or using the cryo-EM facility that has recently started within the department itself. We understand the surface topology. We map the pockets, hotspots, pockets and cavities, and we look at the druggability score of each of these targets. And finally, we depend on the program called Fragment Hotspots, which is hotspot maps, which is a more focused approach towards mapping the apolar, acceptor, and donor regions within the surface of the protein where a small molecule can potentially bind. These are some of the potential drug targets that we have seen, mainly from the arginine biosynthetic pathway and those of the phenol glycolipid, which is on the surface of Mycobacterium leprae, PGL1 phenolic glycolipid, which has antigenic features and is also a glycolipid that's present in the cell wall of Mycobacterium leprae. It's known to have activity towards leading the bacilli into the cells, so infectivity. So blocking this could block infectivity and also prevent the bacteria from producing intact cell wall. So these are the two major areas. There are others that you can see here, like the PAN-C and CoA, which are a part of the fatty acid biosynthesis pathway, and pyrimidine. There are other like PyD and RibH and others which are actually involved in pyrimidine biosynthesis. There are certain TB targets that we have recognized in leprae, that like that of INHA, but of course functionally their relevance is much less when compared to those that block phenolic acid biosynthesis and arginine pathways. So these are sort of potential targets we have recognized with all the pipelines that we have developed in the lab. This is the drug discovery approach, which conglomerates both the computational as well as experimental drug discovery. I'll just take you through the stages very quickly. The first one is target identification, and as I have talked through the bioinformatics part of it, we use biological tools, understanding of the genomic features, to really narrow down onto the number of proteins that we will be able to actually solve the structures. We then do the basic laboratory experiments, as many of you know, like cloning, protein expression, and purification. There are challenges at this stage as well, but at each stage, challenges are met through modifications in the experiments that we do. And then we start off by screening a library of fragments. So where have we got these fragments? These fragments are actually broken down from millions of drug-like molecules or drugs into a very small library size, which is at around 996 small molecules, which contains the core components, the fragments of any drug that can be made. So this has been done by Tom Blundell and Chris Abel, a scientist at the Department of Chemistry at the University of Cambridge, over a period of 20 years. So it's a slowly evolved library that has been stocked within our departments now for us to use to understand the probability of this small molecule to bind in the binding site of the protein. So the idea is to fit in a small fragment-like molecule into the binding site of the protein or into the active site of the protein, study the complexity of the chemical interactions within the biological space, and then expand the interactions, expand the molecule within the binding site, considering the biological properties into a full-blown drug. It's an iterative process. So what we initially do is we do a ligand-based NMR, we do SPR, surface plasma non-resonance experiments, we do thermal shift assays, we do a mass spectrometry to understand the integrity of the protein that we have purified, and then we start by solving the structure with this small molecule. So we start the apomeric structure, we solve the apomeric structure using X-ray crystallography, and then we put in the ligand, we soak the ligand with the protein overnight, and then we solve the structure. We sometimes co-crystallize as well, if that's possible. Then we also look at the affinity of the small molecule to that of the protein using isothermal titration calorimetry and other different functional assays. Once we have this basic information with the fragment, then the fragment actually enters into this cycle, which is stage 4, called as elaborative expansion, where we add a functional group, we expand, and then we continue to add additional functional groups, expand within the biological space, looking at the relevance of interactions it makes and the tolerability of the protein to take up this drug. So it's literally expanding the small molecule within the active site of the protein. So it's a structure guided based on the structure of the protein. The fragment, which is in the active site, is expanded. So structure guided fragment-based drug discovery. We started to work with a very simple protein, which many of you knows in prokaryotes, which is FTSZ, filamentous temperature sensitive Z-like protein, which forms a Z ring at the center of the bacteria during its cell division and separates the bacteria into two. Blocking it can actually block the cell division and can lead to the death of the cell. So we started off working on this, which has been a target for many other bacteria as well, just to understand if the concept of fragment-based drug discovery can really be applied to any of these basic proteins within Mycobacterium leprae. There is some thermal shift data on the fragments that we have collected. At least 2 of them showed significant shifts, which are promising. We are in the process of expanding them into more elaborate compounds. We did CPMG and standard NMR spectrum experiments, and have noticed the NMR shifts as well. And then this is how it is. Like within the protein structures, we have the GDP bound states. You can see the GDP molecule there. We would really like to displace this GDP that actually helps in the polymerization of the protein and fitting of the formation of the Z ring to a state where we have a small molecule that occupies that place. So it's a very small Turing structure that you can see that is sitting very closely when superimposed with GDP. It may not be able to displace GDP right away, but as it's expanded in the chemical space, in the biological space, it can do that. And at a point, we'll be ending up with a molecule that actually displaces GDP and blocks the function of FTSZ. There is another molecule similar to that, which I think you can see here. This is the simple overview of the robotics that we use in the lab. Many of you might know about these, the basic dispensing dragonfly on the top, the mosquito that actually coats protein onto the plates, crystallographic plates, and then there is a formulatrix machine that actually preserves the plates at different temperatures and takes photographs automatically and sends them to the online portal that we have, so we can log in from anywhere and look at the crystals as they grow. And once the crystals are grown, we can actually take them out of this chamber and start fishing them to send them to the X-ray plants. This is the diamond light source, the synchrotron facility that we have close by. This is a medium wave synchrotron that has it's quite helpful for biological research, although half of it UK has committed to space research. And each of the scientists were given these hutches, which are automated crystal placing robotics or crystal shooting robotics that are there. So we direct our beam lines to these hutch. Beam lines are actually directed to these hutches, and we place our crystals. This robotic arms actually picks crystals from our plates and puts them onto the beam path so that it's shot and the data is collected and processed. So the right side is a picture of me working at the synchrotron facility recently. Just to continue on what we were doing with the cell wall synthesis blocking at this stage. This is my colleague Martha from Spain, who is working on this project as well along with me. We are trying to look at this phenolic glycolipid pathway and block its proteins so that will block the bacteria from actually entering the nasoepithelial cells and also entering the Schwann cells of the nerves. So Mycobacterium leprae has the property of entering the nerve cells and blocking destroying the nerves so that the sensation is lost and the deformity occurs in the limbs because of nerve damage. So it's a polyneuropathic situation that Mycobacterium leprae induces. To prevent that from happening, so we are blocking Mycobacterium leprae's essential proteins that prevent it from entering the nerve cells as well, along with the other cells in the body. This is the basic biochemistry around the phenolic glycolipid sugar moiety that's there, which is slightly different from that of the tuberculosis, and of course largely from M. bovis. And there is you can see that there is a trihalose monomycolates that actually join to the phenol thiazerol the entity that carries the phenolic glycolipid sugar glycosyl moiety of it. So this is the whole PGL molecule, and then this is the basic biosynthetic pathway. We are looking at the FAD-D32 step. I'm not sure if I can highlight my. Yeah, there's no possibility to show with a cursor, but if you can see the condensation reaction that I highlighted on the left side of the screen, you can see that FAD-D32 is an enzyme, fatty acid adenylating enzyme, which is helpful for the activation of mycolates to myromycolates, and then through condensation to mycolic beta ketoesters with the formation of complex with PKS13, polyketide synthase 13. So these are the potential drug targets that different groups from around the world are working on. There is a similar scientist in the US by name Professor Jim Sakatani, who is working on the PKS13 for tuberculosis, and I'm working on FAD-D32 from leprae. Maybe both of our findings can be integrated together over a period of time to see if we can have similar set of compounds that can inhibit the functions of these proteins, both in tuberculosis as well as in leprae. But that's the direction, and these are the potential targets that we are working on now. There's quite a lot of literature that has been on this protein and the small molecules that actually inhibit the function of this particular protein. We are in the process of doing some sort of virtual screening to understand the analogs of the diaryl coumarins that can actually join this active site of this particular FAD-D32 and block the function. So that's still going on. These are structures from different bacteria. We have a model from leprae, which is bound to the ATP molecule that's there that you can see. As you see the protein structure, you also see these yellow, red, and blue color patches. These are nothing but the fragment hotspots. Our program maps them onto these proteins and patches them on the structure so that we know when we are binding small molecules, what we are binding where, and how stable those interactions can be. This is available online. If you type fragment hotspot maps in Google, the first link that you get is the program that's been developed and been used widely around the world. As I was talking to you about HARP, I just wanted to show you the pipeline for HARP that we developed. This is the in silico saturation mutagenesis, maybe relating to what we are experiencing in COVID-19 now, that a mutant occurs one after the other. So, what would be the impact of such a mutation on the structure, on the small molecule binding, on the ability to induce immune response if we are thinking of a vaccine, and things like that can be studied if we adopt an approach like this. Nothing but it's not an experimental saturation mutagenesis, this is in silico saturation mutagenesis, where we mutated every residue within the drug target proteins of Leprosy to all the other 19 amino acid types and measured the impacts. In reality, this cannot occur because of the ambiguity of the genetic code, and of course the nature of the genetic code, in which multiple codons code for only single amino acids. So, but just to have this iterative knowledge on the possible evolution of strains that can harbor these mutations, we have done saturation. So it's a tremendous amount of work. Here, just for one subunit, you can see 21,394, but in total we have generated around 122,000 odd mutations for the entire structures of three drug targets in Leprosy. HARP is available online. If you just type harp-leprosy.org, you should be able to access the database and study the impacts. This was the original work was published in 2018. Then later we published the saturation mutagenesis in 2019, and then followed by the HARP database early last year in 2020. So this is one single subunit. We mutate each residue to all the other 19 residues and then map the impacts, like the average or the maximum destabilizing impacts that each mutation can have. So from these maps, you can understand that the red regions are highly destabilizing. They're not really sites for small molecule binding, because if the bacteria induces a mutation in that region, it can lead to possible loss of drug interaction with the protein. So then what we thought was, why don't we work on the converse of it, like the neutral regions, or the regions that are less impacted, and then we map them as blue there, because they can also be as safer regions for small molecule binding. So we looked at the conservation scores, how conserved each residue is, this stabilizing impacts, or destabilizing impacts, and neutral effects. We looked at the interactions, how these interactions between proteins can be impacted if a mutation induces. We looked at then we started to overlay our hotspots to that of the mutation cool spots, or regions that are safe for us to hit the bug with a small molecule. We have seen at least two regions where our fragment hotspots are overlaying with the mutation cool spots. So if we design a drug that fits into this particular area of the protein pockets, we shouldn't be able to have a major problem with mutations if the bacteria induces mutations, because they don't have a large impact either on the protein structure or on the ligand binding. We did random large-scale simulations. I won't really get into more details of this, but we were able to do sort of a microsecond level simulations with large structures using the HPC systems at Cambridge to really understand the stability of these interactions of rifampin with the RNAP in Mycobacterium leprae. Let me just move to the next one. We had, whenever we develop tools or when we develop techniques, we really wanted to see the correlations with the other programs that are existing within the field. There's not quite a great correlation that we have seen with the other programs, given the nature of our approach of saturating the residue to all the other 19 residues, while most of these programs have focused on only existing naturally occurring mutations. So these are basic observations which are available now on a published literature. You can see them, and HARP is available online. One can actually browse through and recognize. With these, I think I would like to conclude my talk. talk. These are the current connections that we have with American leprosy missions. The government of the UK has offered us access to Diamond Light Source and the Science and Technology Facilities Council, which is a part of the UK Research and Innovation. We also strongly collaborate with the Cambridge Crystallography Data Center, CCDC, from where the gold docking software has been developed. Some of you may have used gold in the past. And then we also have this XChem fragment screening platform that's available, which actually helps us to soak our crystals with not just one small fragment, but with approximately 300 to 400 fragments in a single plate, and then screen using crystallography in few hours of time. So it's a recently developed beamline that's set up at the Diamond Light Source that I showed. And other collaborators from the University of Melbourne, University of Rutgers, and University from a Brazilian university in Fiocruz, in Minas Gerais, have been collaborators on the mutation analysis. Thank you very much for your time, and it's my pleasure to take any questions that you have. Yeah, there is one question that I can see. Sorry, Mr. Manish, Dr. Manish Kumar, if you'd like to speak, yes, I can respond to that question quickly.
Yes, sir. You can respond to that question, sir.
Okay, so there are, I think, few questions as I see from the top. May I know what this proteome means? As a proteome, in the context of bioinformatics or computational biology, is the set of all proteins, all gene products that are existent and functional within the bacteria. So that's the definition for the proteome. And there's another follow-up question. There's another question from somebody on why does leprosy attacks the nerves and fingers especially? To be honest, we don't know why that happens. They have phenolic glycolipid, which has got a very characteristic predilection to the human Schwann cells. Bacteria normally survives by actually building its thick cell wall, cell coat, which consists mycolic acids. And Schwann cells within the nerves contain quite a lot of cholesterol and apolipoproteins. Bacteria tries to harvest this cholesterol using apolipoproteins of the host from the Schwann cells. That's one theory in place. There are markers that have been determined by us and by other groups to say so. And there were cellular studies that showed how M. leprae enters the cells. But why it specifically chooses nerve cells to go and sit there is something we really don't know. And we are in the process of exploring. Yeah, high resistance and high level of susceptibility. So I think these are two different aspects. I might have overlapped the statement. The reason is high resistance is there are certain pockets. One of the characteristics of leprosy transmission is presence of endemic foci. So there's a focal transmission of leprosy. So with the same focal mode of transmission, there is also a focal mode of transmission of resistant bacteria. So these pockets are named as highly resistant pockets. So it's an attribute to a population and not to an individual or to the bacilli. It's more attribute to a population. And high level of susceptibility is that the contacts are more prone to leprosy infection when compared to non-contacts of leprosy people. So it's very difficult to quantify that susceptibility, but in a population term, the contacts are a population that are highly susceptible to leprosy. And that's what is meant by those slides. Which drugs are resistant to Mycobacterium leprae in the present trend? Dapsone, rifampin, and ofloxacin are known to be resistant to leprosy. We have only one strain of clofazimine-resistant leprosy propagated in Vellore, but we don't know if that's a significant problem yet.
So thank you, Sundeep sir, for your presentation. Presentation related to leprosy, and So we are very happy for answering the questions which were posed by the students. And it's a great presentation, sir. And thank you for sharing your knowledge, which is related to the developing of web source with proteomic model, as well as the impact of mutations and drug targets, and about the Hansen's database. So we are very thankful to you for being a part of this international e-conference on March towards drug discovery and drug development, sir.
Thank you and all the very best. And thank you for inviting me too.
So now I request Dr. Ragini, HOD, Department of Pharmacology, to give oath of thanks to both of our speakers as well as the participants.
Good morning. Dear participants, speaker and management, it gives me immense pleasure to deliver a vote of thanks for this event to all the dignitaries assembled here. I would like to thank our speaker, Krishnamraju Ankireddy, PhD, Research Scientist USA, on delivering the seminar on the synthesis and characterization of the nanoparticles and their application, and Dr. Sundeep Chaitanya PhD, Director of Research and Innovation, University of the Cambridge, on the structure-guided fragment-based drug discovery for leprosy, by sharing their research knowledge with us. A special thanks. I would like to thank our beloved principal, Dr. Lakshmi Narasimhaya, organizing secretary, Dr. Swarnlata, organizing committee, registration committee, and volunteers, and the last but not least, all the participants making this conference a grand success. Once again, I thank one and all present here. Thank you very much.